Right. Good afternoon, everyone. Is everybody doing okay? Not scienced out yet, hopefully. Normally, if people have been to convention, typically we were really focused on research on Saturday. We have our research forum and our clinical trials showcase. This is an evolution. You're seeing what's happening in HD science. It's true. There are more and more companies coming to this space, more and more researchers coming to this space. There's too much great research going on in HD to just have it on Saturday. So we've created this new session, a Huntingtin lowering session. This is the first because there are so many wonderful things going on. You'll hear about some tomorrow, but you're going to hear two very novel approaches that are being undertaken by two companies, both actually in the Boston area, to tackle Huntington's disease. One, a silencing approach using oligonucleotides, and then one using viruses to deliver drugs into the brain that will also attack and lower Huntingtin. But our first speakers, we're doing a tag team presentation from Wave Life Sciences. First speaker is going to be Wendy Erler. Wendy is the Senior Director of Patient Advocacy and Patient Engagement at Wave Life Sciences. In this role, she leads all patient advocacy efforts for the company. And as an early stage preclinical company, they really value the importance in getting this right. I was amazed when this is a brand new company. And when did you start? In January. They have a director of patient advocacy. This doesn't happen in the biotech world. So they're really, truly committed to working with the patients. And Wendy's hiring is evidence of that. Wendy's previous experience in the rare disease included working in ALS and SMA and hemophilia for Biogen, where she was before joining Wave Life Sciences. She earned her MBA from St. Joseph's, and she'll be kicking off the presentation. And then she'll be handing it over to Roberto. Roberto Garciolini is an MD PhD. He's a senior VP and head of early development at Wave. Previously, before joining Wave, he's had roles including Vice President of Emerging Business Unit at Shire, was Senior VP of the Pharmaceutical Development at Dicerna Pharmaceuticals, a company that he actually co-founded. He was the Chief Medical Officer at Cerna Therapeutics, and he received his medical degree and board certification in internal medicine from the University of Perugia. Did I say that right? Yep. Medical School in Italy. He completed a postdoctoral fellowship in clinical pharmacology at the Mayo Clinic and is certified by the American Board of Clinical Pharmacology. So I'm going to turn it over to Wendy and Roberto so they can give, you'll hear a little bit about their allele selective approach to lowering mutant Huntingtin.
Thank you, George. Thanks to all of you. It's such a privilege and an honor for us to be at this meeting and have the opportunity to engage with the HD community, patients, caregivers, physicians, social workers. I can't tell you how much it means to us. And as George mentioned, I joined Wave because of the early and important commitment to patient advocacy that's evidenced by having a seat at the leadership table in an organization this early in its developmental stage for clinical therapies for Huntington's disease. So when we say we want the patient voice at the table, we really mean it. And I like to say we want to walk the talk. So this is a little bit of a background on Wave itself, just to level set that, we're a new preclinical company, but we've actually been around since 2009. And through a couple of different steps of evolution of the company, it brings us to today. And the exciting news and what's relevant to all of us in the room is we plan to file our first 2 INDs, that's the first step to getting a clinical drug through the FDA process. So we're going to file our first 2 INDs by the end of this year, both of which are in Huntington's disease. So you'll hear more about why we have 2 and what that means. But this is real and tangible, and we're really anxious and excited to work with all of you and do something very meaningful. So some of the numbers on here show financing, and the reason that's important to all of us in the room is if we have some financing, we can get these drugs through the clinical trial process. So we're in a really good stage to be able to do that. Our commitment to the HD community is real in a lot of ways. Not only do we want to bring these INDs forward and get drugs into the clinic and execute the clinical trial process, we want to do it in the right way. And so hearing from you about what's important for disease modification and symptom relief, and what sort of life changes do you all need that are really meaningful, that mean these drugs work, we need that information coming in from you, because if we sit around the table and try to guess, we may not know what's truly important. So that's a big part of my job, and patient advocacy is a distinct, meaningful priority within our organization. We also, as part of our commitment to you, have a commitment to transparency and open communication. So as soon as we can share information, we will. That doesn't always come along on the time frame that everybody in the public wants necessarily, but the commitment is the minute we can share information or we have any data updates or we're going to issue a press release, you guys will be hearing about it in real time. And that commitment often is executed through organizations like HDSA, but it's also, there's an opportunity, there's an email at the bottom of this slide. You can email us and say, what's going on? What can you update me? And that's that dedicated patient advocacy at WAVE. It's actually wavelifesciences.com, so I'll update that and get it out to everybody. We're building really close ties to the Huntington disease advocacy group, so HDSA as well as the chapters and other groups, because that's our way to hear from you all collectively. So for everyone that's involved with their local organizations and doing the walks or fundraising or whatever you do to impact your community, know that it is even more real than that. Everything you share with your chapters comes back to organizations like ours, and it gives us a chance to empower our trials and empower our leadership team to make decisions that are meaningful for everybody in the HD community. Also, I think it's important to know, and we heard a little bit about this in some of the sessions earlier today, we are learning and have cross-learning opportunities from other, specifically rare disease spaces, other neurological diseases as well. But the rare disease spaces in particular, individually, HD's a small rare disease, it's hard to be heard. Collectively, rare diseases are a really strong, powerful voice, both legislatively, in the trial space, with the FDA, and all sorts of other constituents. So making sure we're open to the opportunity to learn from other rare disease spaces and how they're moving advocacy forward, how they're moving science forward is really important for all of us. So I'm going to turn it over to Roberto, who's going to talk to you about what we're doing at WAVE from a science and a clinical development perspective.
Thank you, Wendy, and thank you, George, for inviting us here to convey to all of you our story, our exciting opportunity in creating for the development of meaningful, impactful treatment for Huntington disease. I'm vertically challenged. I would like to move away from the podium and be closer to you, but apparently camera requirement asked me to be here, and so I'll be here, and I hope that I convey equally well also with the 10% of myself here. The commitment to, sure, that's one point. So, the commitment to neurological disease is represented here by our pipeline. We are totally vested in utilizing our chemical platform for neurological disease, particularly rare disease, genetically determined disease. And you can appreciate that our commitment to Huntington disease is actually represented by the first two programs that are going towards IND filing at the end of the year. We'll be committing in the next 5 to 6 months to fully execute the requirement, the preclinical work required by the agency in order to move into clinical experimentation. And such experimentation is expected to start early in 2017. We can also appreciate that we don't stop with Huntington, but we actually have other areas of neuromuscular diseases as well as neurological diseases. And these are commitments that over time will become towards the clinical conduct of clinical trials. In order to convey our story, we'll cover a few concepts that might or might not be familiar with you. Some of them have been already covered by Dr. Wild in his very explicit presentation in terms of antisense, oligonucleotide, and gene silencing. But other will be the chirality of the oligonucleotide that is our chemical platform. The novelty will bring to the place, and more importantly, the selective silencing of the mutated Huntington disease gene without touching the wild type or natural allele. So let's go over this a few points. Dr. Wild, as I mentioned already, now the mechanism of antisense oligonucleotides, which are molecules that match the messenger RNA of a transcript, and matching and destroy them, engaging RNA's enzyme, impede the production of the protein. In this case, is SOD1 protein, but in the Huntingtin disease would be the Antisense protein. So we know for the past 25 years the antisense oligonucleotide work this way, silencing unwanted protein productions at the level of messenger RNA. Now, we also know that for 25 years up to today, and even on the market, mipomycin, one product on the market, antisense oligonucleotides are mixture of chiral differently isomer. A crash course in chemistry. Antisense oligonucleotides are stabilized utilizing phosphorothioate. Anytime you introduce a phosphorothioate, you produce two isomers, two specular images of each other. They are the same sequence, but they are not overimposing each other. So they are different molecules, actually. And when the number of chiral centers, like in this compound, which has 19 chiral centers, is large, your resulting compound mixture is 500,000 compounds, all of them different and randomly distributed. Some of them are good, some of them are very active, some of them might be neutral, some of them might be toxic. So that's the status of the art for until 2 years ago. 2 years ago, our scientific inventor came up with a process that controls chirality at the chemical synthesis level. This is our platform. This is the difference is from a state-of-the-art mixture here, 500,000 compound. Through our platform, we are able to develop a single isomer oligonucleotide optimized for a variety of activity. What we optimize using chemical chiral control is the stability of the oligo, is the activity or potency, is the lack of immunostimulation, and importantly, the specificity, the selectivity between a mutant allele versus a normal or wild-type allele. The result is an optimized isomer that could be applicable to a variety of modalities in oligonucleotides. The antisense, which is the most common and which will be the subject for today, but also the RNA interference, as well as the exon skipping approach. So that's our platform. With chirality comes also a very important characteristic, which is selectivity. We are able to direct the cut of the messenger RNA at one point and one point only. And we can take advantage of particular modification in the messenger RNA, like SNP, or single nucleotide polymorphism, to actually make the cut on the intended and intended only sequence. There's an example here where on the assay, or RNA-chase assay, a mixture of oligos are potent in knocking down or silencing both alpha allele and mutant allele of Huntingtin disease. But when we use an optimized stereochemistry, able to discriminate between by a single base, we actually have a great deal of separation between the silencing of the intended target, the mutant, and the sparing or untouched of the wild-type allele. We believe these are very important medical consideration, because clearly this is the intended silencing, and we want to maintain the wild-type production of Huntingtin. We don't know what the potential consequence of silencing the wild-type Huntingtin will be on the long term, while we know that this is the intended reduction of the Huntingtin protein, the protein that produces the disease. Additional characteristics conferred by the stereopurity of oligonucleotides are represented by another two features. One is the lack of immunoactivations. Complement activation has been involved in a variety of neural neurological, pathological conditions. And actually tomorrow, Dr. Saltarelli will educate us on the opportunity to use complement as a target for neurological condition silencing, specifically Huntington disease. You can see that while the mixture oligonucleotide has a substantial capacity to induce immune activations, our candidates is totally devoid of that. So chirality, devoid of any immunostimulatory activity our candidate, without paying a price on reduction of protein binding. Protein binding is a characteristic of all the oligos, is a key element for circulations, both centrally and in the cerebrospinal fluid, and also a facilitator of distribution of the oligonucleotides. So, what is our approach utilizing this platform for Huntington disease? We are going to exploit two things. One, this selectivity that I already mentioned to you and described to you, and the presence of single nucleotide polymorphism in our genome. As single nucleotide polymorphisms are very common, and they are all across the genome, the human genome and also animal genome, and usually they are innocuous, they are inconsequential. But when they co-segregated with, for example, the mutant Huntingtin protein, and close to the CAG repeat, which is the actual cause of the disease, they could become an attractive target for silencing, right? So we can exploit the presence of this SNP as well as selectivity to cut the messenger RNA right there and to inhibit or actually abolish the production of Huntingtin protein. So we believe that this approach has a disease-modified opportunity. We block and we stop the production of Huntingtin protein, and we hope that that will result in the stopping the progression of the disease. Why single nucleotide polymorphism? There is a vast body of literature in the past 20 years describing the presence of certain number of SNPs across populations, across geographic location in patients with Huntington disease. And such frequency has been characterized mostly on European descent, American, Canadian, and partially South American. We know that the most frequent SNP associated with the CAG repeat is about 40%. If we devise a treatment targeting this SNP, will cover about 48% of our patients. If we devise two treatments, our percentage goes up, and we can cover 2/3 of the patient population, and so on. We can go on to develop a SNP3, a SNP4. So that's our approach, to provide two solutions for very personalized treatment of Huntington disease, exploiting selectivity and single nucleotide polymorphism targeting. This slide is the final slide, and I hope this is going to be the take-home message. We selected two SNP targeting therapy for our first two IND in order to cover about 66% of the patients with Huntington disease. The clinical trial that will start in 2017 will be conducted in parallel, and the patient enrollment in the trial will be contingent upon the positivity and the association of the SNP with a blood test. So patient as a part of the screening will take a blood test, and the SNP characterization will be identified. SNP1 patient will go on trial 1, SNP2 patient will enroll in trial 2. The clinical trial will be conducted in both the United States and also abroad, and enrollment is due to start early next year. Of course, these are safety trials, so a word of caution, we are in the preclinical stage. We have 6 intense months to complete our preparation for IND, and the first clinical trial will be of safety nature. Nevertheless, the excitement is very high. Thank you.
We have time for questions. Does anybody have questions for Wendy or Roberto? Yes?
How is this SNP modification to be delivered in these trials? Is it orally across the blood-brain barrier?
A great point. And oh yes. So the question is how the oligonucleotide, SNP-specific oligonucleotide, will be delivered to the patients. These are large molecule, and as Dr. Wild mentioned to you, and mimicking to a certain extent what the ongoing clinical trial is with non-selective antisense oligonucleotide, the route of administration will be intrathecal. Intrathecal administration, spinal tap, lumbar puncture. Intrathecal administration has been demonstrated to produce profound and diffuse distribution of the oligo into the brain as well as into the spinal cord. The brain, of course, is the target for the intended therapeutic benefit. Yes?
Are you going to do any, or have you done any preclinical work in mouse or other animal HD models?
The question is whether we have done some preclinical work in model of Huntingtin disease. The human SNP, the target of our program, is not certainly present in animals. Nevertheless, there is an attempt to generate transgenic mice carrying the two SNPs I mentioned to you, and actually a third one that could be the third target for us. But at the present time, we don't have those data available. There was a, oh, go ahead.
Is it anticipated that you'd have to give these SNPs one time or repeat the dosing over time?
The question is whether the treatment will be a one time, like gene therapy, or will be a repeated time. These are oligonucleotides, so they are silenced in the target on a periodical basis. Let's go that way. And at the present time, the intended frequency of administration is 4 weeks. But we will know more from preclinical work ongoing as we speak whether those paradigm is right or not. In other words, is the resident time of the oligo in the brain and or the pharmacodynamic effect, the protein lowering effect, is longer than 4 weeks. Certainly, they will suggest eventually using less frequent administrations. The opposite is true if after 2 weeks we don't find oligo in the brain and we don't see a sustained silencing of the intended target. On the back.
How will you be selecting your sites for your studies? Will you be going for HSG, or how would one be
Sure.
considered
Yeah.
to be a site?
The question is whether what would be the criteria for selection of clinical site for the two trial. Again, these are two trials going in parallel. So, and we are being in the process right now in terms of clinical operation to evaluate a variety of sites, mostly the United States, but also start peeking into the European as well. The criteria right now are large volume site, because this is a short study, short-term, small number of patients. So large volume of site will be the best to accommodate both the selection criteria, the SNP selection, as well as the number of patients we intend to enroll. Yes?
Your comment about broad coverage, I know you showed a slide that had, number of SNPs. Yeah.
Right there.
Are you saying broad coverage 48% or closer to 80%?
I say that the first two interventions, the one we are working right now, give us coverage of 65%, two-thirds of the population. We don't exclude the opportunity, as you have seen on the pipeline, they will go after SNP3, which will add another 12% of patients. We are learning actually from one to the other. In other words, we are learning from SNP1, which is the most advanced one. We learn a lot of rules in terms of synthesis, in terms of characteristic, and now applicable to the second SNP, and the third SNP development will be accelerated on the basis of what we learn. There is one hand there.
Will you be monitoring the levels of the wild type protein as well?
The question is whether we'll be monitoring the level of wild type protein or mutant protein.
The non-mutant protein.
Okay.
There's no interference in that level.
Certainly. So in the multiple dose section of the trial, we're going to have monthly administration. And any administration will be preceded by the collection of spinal fluid. And the spinal fluid will be the subject for measuring both the wild type and the mutant protein as a surrogate of target engagement and silencing of the intended target, the mutant one. Right in the middle.
Once this trial gets up and running, do you anticipate it being randomized controlled, or will everyone?
I'm sorry, I can't hear you.
Will it be a
Do you
randomized
anticipate
control?
this trial, once it gets to human safety studies, to be randomized controlled, or will everyone get the intended snips?
Okay. So the question is whether the trial will be randomized and placebo-controlled or open label or everybody use treatment. The current design, which is still in the discussion with investigators and clinicians, are for a placebo-control approach, minimize the number of placebo. In other words, the ratio could be 4-to-1 or 3-to-1, but really having a placebo control as a reference point, because ultimately this is a safety study first. On the back there? I need somebody repeating the question for me this time.
She just wanted to know if you had clinical trials before, whether you would be considered for this one.
So the question is, if you've been part of an experimental trial before, there will make you automatically selected individual. There are two ways to answer to this. One, if you have been involved in a clinical trial, experimental trial with some other medicines, you would be eligible to enroll after 6 months from the end of that previous trial. We don't want contaminations. But if you are a SNP1 positive subject and you go on this single ascending dose phase of the protocol, you are eligible to go on to the multiple dose phase of the protocol, which after will be the one with more chances to be beneficial for any changes on the protein as well on the symptoms. Is that satisfactory? Oh. Okay. The question is about stem cell research, and I'm not qualified to talk about those because it's not the area we are operating nor have experience on it. So the question is about marijuana oil, and I think I stick with the previous response.
Thank you, Wendy and Roberto. I didn't know there was marijuana oil. Don't look into that. All right, I'm going to pull up. I see wave. Is yours on here? Okay. Don't see it. don't see the mouse now.
Oh, did the mouse
I don't see them. Oh, there it is.
It's on the desktop.
Okay, let's go.
Just close there, so.
Oops. Okay.
I know the mouse is
Oh.
crazy.
All right.
There you go.
Okay. All right. All right, last talk of the afternoon. So our final presenter of the Huntingtin lowering session is Dr. Lisa Stanek. Dr. Stanek is a senior scientist in the neuroscience group at Sanofi Genzyme in, I guess it's still Framingham, Massachusetts. Dr. Stanek received her bachelor's degree from Union College and her PhD in neuroscience from Emory University in Atlanta. She trained as a postdoctoral fellow at Harvard and Mass General Hospital, where she performed research on the role of microRNAs in synaptic plasticity and cognitive function using different transgenic mouse models. She joined Genzyme as a staff scientist nearly a decade ago in 2008 to work on neurodegenerative disease initiatives including HD, SMA, spinal muscular atrophy, ALS, and Parkinson's disease. And she's currently evaluating gene-lowering technologies for the treatment of HD. Presently, Lisa is responsible for leading the HD drug discovery research project at Sanofi Genzyme. So I want to welcome Lisa to the stage where she's going to talk to you about AV-mediated gene silencing for HD.
Thank you so much, and thank you so much for George and the organizers for allowing me to come here today. This is my absolute favorite HD meeting of the year. I really look forward to it. So I love engaging with the community and the patients and their families, and really look forward to your questions. So like George said, I'm a senior scientist at Sanofi Genzyme, and I'm working on an AAV-mediated gene silencing approach for the treatment of HD. So for those of you that went to Ed and Jeff's Ask the Scientists Anything, you probably already are very familiar with this, how Huntington's disease works and the gene silencing approach. So you've got, is there a pointer here? So you've got the mutant Huntington's gene, which codes for a mutant Huntingtin messenger RNA, which creates the production of a toxic mutant Huntingtin protein, which causes all the symptoms of the disease. So if you could stop the production of the mutant Huntingtin protein, you would obviously have transformative therapeutic benefit to the patient. So that's where the gene silencing therapy comes in. So what is the rationale for these Huntingtin lowering approaches? The preclinical data package supporting Huntingtin lowering is very robust and supportive of transformative therapy. So more than just palliative therapy where it will treat just the symptoms, this is a transformative therapy that could possibly prevent or halt disease progression. So this therapeutic concept of lowering mutant Huntingtin has been very well validated using animal models, primarily mouse models of the disease, using a number of different modalities. So I know Ed and Jeff talked about flavors of gene silencing molecules. There's the Antisense oligos, which they just spoke about from Wave Therapeutics, and there's also Ionis working on ASOs. RNAi gene silencing that I'm going to talk to you about today. And there's also some newer approaches, the genomic editing approach. This was kind of a hot topic in Jeff and Ed's session, where people are now using zinc fingers, or you may have heard of CRISPR-Cas technology to actually edit the genome. This is probably less far along than the ASO and the gene silencing approach, but nevertheless it's really exciting and it's really emerging. But using all of these modalities of lowering mutant Huntingtin, you've seen very robust transformative benefit in the mouse models. So the preclinical testing of this therapeutic concept has shown the correction of biochemical aberrations as well as significant improvements in motor and behavioral deficits in these animal models of the disease. So at Genzyme, we chose to evaluate our therapeutics using the YAC128 mouse model of Huntington's disease. So I don't know how familiar you guys are with mouse models. There's probably up to now, 20 or more different transgenic mouse models of Huntington's disease, and we've selected to use specifically the yak model because these mice show a striking similarity to the humans in terms of their anatomy, physiology, and genetics. These transgenic mice express the entire human Huntington's gene with 128 CAG repeats. Some of you may have heard of these fragment mouse models like the R62. That mouse model just has the expanded CAG repeat, but it doesn't have the entire gene with introns and exons. So this is really a very genetically relevant mouse model because it has the entire Huntington's gene. The animals show early electrophysiologic abnormalities in terms of the behavior of their cells in the brain. They show nuclear inclusions, or these Huntingtin aggregates that's also found in the human brain, and they do show neurodegeneration. These animals exhibit functional deficits, so motor, cognitive, and psychiatric phenotypes, which I'll talk a little bit more about later. And what's a very unique feature of this mouse model is that the older mice, about 12 months or older, they exhibit selective degeneration of medium spiny neurons, which is exactly what happens in the human condition. Mouse models tend to be somewhat protected for reasons unknown to neurodegeneration, so that's why we really have to push the system with 128 CAG repeats, that's clearly a lot, in order to get them to manifest this disease. So here's just a snapshot of how we conduct all of our preclinical testing studies at Genzyme for Huntington's disease. We've got our intervention up here, which is, in our case, we are injecting a virus into the mouse brain. We then obviously let the animals to recover and heal from the surgery, and then we measure some kind of functional performance. So in this case, it's motor performance. This is a rotarod test where you put the animals on a rotating rod and you measure how long it takes them to fall off. A Huntington's mouse will obviously fall off sooner than a wild type or healthy control. We also have tests of psychiatric function in the mouse, and this is called the Persult swim test, where you can test for a depressive phenotype. Obviously, these mice probably are not depressed in the way we think of depression, but we call it a depressive phenotype because it can be reversed with SSRIs and antidepressants. So it's just, I don't like to anthropomorphize and say our mice are depressed, but it is a way to sort of measure that type of psychiatric phenotype in the animals. So sometime after the functional testing, we actually sacrifice the mice, collect the brain tissue, and do a number of different biochemical and histopathological analyses to see how our treatment affected the brains of these animals. So I said the approach that we're undertaking at Sanofi Genzyme is this AAV-mediated RNAi or RNA interference approach. So I don't want you to get bogged down in all the details of the slides, but to say that RNA interference is actually a natural process that happens in all of our brains. It's a way that our brains can sort of cull out, messenger RNAs and proteins that they're not using anymore. So we're really, as scientists, we're hijacking this process, and we're putting in RNA interference molecules that will code to tell the cell to get rid of the Huntington's protein. So these RNAi molecules can come in a number of different flavors. There's the artificial microRNA, which we are using, and there's also siRNAs and shRNAs. You may have heard of these as well. It just depends how processed that RNAi molecule is. We actually we prefer to use a less processed RNAi molecule, which is this artificial microRNA. It requires the cell to actually do some processing itself, as opposed to putting in the final product, which is like an siRNA. And this really helps with toxicity, because if you put in the final product, it can overwhelm the cell's endogenous machinery, and it can lead to some toxic issues. And we've seen that in mouse brain studies and even primate studies. So we like to use a more natural endogenous RNAi molecule known as the artificial microRNA. And how do we get these things into cells? You can't just, put them directly in the brain and have them, do their job for a long period of time, because they just get digested by the enzymes in the brain. So we're going to propose delivering these molecules using AAV or viral vectors. So AAV viruses have been used for some time as a tool for gene silencing. These viral vectors are replication defective parvoviral vectors. They transduce non-dividing cells, which is absolutely critical for a CNS intervention. We need to transduce neurons. A lot of viruses don't do that, but AAV viruses specifically do. They're non-pathogenic, so they don't cause disease. A very important point in the world of biologics is that we can actually make these things in large enough quantity and good enough quality to treat the Huntington's patient population. So, you know, we have to generate clinical grade vector that's in enough quantity to treat the patients that we're looking for, and that's actually not something to be minimized. It's something Genzyme's been doing for a really long time. We have a lot of experience in it, and we're very comfortable with generating these viral vectors in, again, a clinical grade quality. And the most powerful part of using the use of viral vectors is that a single intracranial administration of the AAV could provide long-term suppression of Huntingtin and long-lasting therapeutic benefit. So you wouldn't have any kind of complications of repeat administration, having to go back, you know, regularly for infusions, or, you know, some people have proposed using pumps to continuously deliver the therapeutic. It's really just a one-shot deal. You go in, you get your injection, and you're done. And it does sound very invasive. It's brain surgery. It's direct intracranial injection into your brain, but these procedures are performed very routinely. This is done very regularly for the treatment of Parkinson's disease, the implantation of deep stimulating electrodes into the striatum. So the actual surgical procedure is quite routine. Most neurosurgeons can conduct it in your average clinic. All right. So now I'm going to show you some of the preclinical data that we generated in our mouse model, in the YAC128 mouse model of the disease. So this is just a construct showing our microRNA, our AAV microRNA. In our early mouse studies, we used a GFP, green fluorescent protein tag. This allows us to see visually where our virus is going to make sure that we're distributing it, enough of it throughout the brain. So we injected our animals when they were 2 months of age. 2 months later, we actually put them on the rotarod and did all the functional behavioral testing. We did the per-salt swim test, and then right after that, we sacrificed the animals, took the tissue, and looked at the brains. What this is illustrating here is where the virus ended up after injection. This is a mouse brain, a cross-section of mouse brain. This is the striatum. So everywhere there is this white color, it's actually green. You see a widespread expression of the virus in the brain. So where we put the virus, it's spreading out, and it's really covering the brain areas that we're interested in. This is showing when we harvest the striatal tissue of this animal, we're getting about 50% reduction in the Huntingtin level. It's important to note that this is a non-allele specific approach. So unlike what they were speaking about for Wave Life Sciences, where you're just targeting the mutant allele, here we get knockdown of both the wild type or the healthy Huntingtin as well as the mutant Huntingtin, but the levels of knockdown are only about 50%. So we're not wiping out entirely all of the healthy Huntingtin in the brain. We're going for a partial reduction. And then here are the results of the behavior testing. So again, we put the animals on the rotarod to test their motor coordination, and the per-salt swim test to test how long they swim versus float. Depressed mice, you actually put them in the beaker of water, and they just give up very quickly, and they just start floating. With the treatment, you put them in the beaker of water, and they swim the entire time, suggesting they're quote-unquote less depressed. But we see significant behavioral improvement with our AAV treatment in both the motor coordination and the per-salt swim test. We also wanted to look at the brains of these animals to see if we've corrected any of the neuropathology. So this is an interesting study because this study had to be conducted when the animals were much older because, like I said, those aggregates don't come online until around 12 months of age. So we injected the vector at 7 months of age, and we planned originally to just sacrifice the animals when they were a year old and look at the brains for aggregates. But because I had the mice in our facility, I said, well, let's take a look at them and see how they're doing. We put them on the rotarod, and to our surprise, these are symptomatic animals. So they're 7 months old. They have Huntington's. They have all the features of the disease. The blue bars represent the animals that were treated at 7 months of age with our therapeutic. You can see that we actually reversed the disease in these animals. So they started out with the presentation of Huntington's disease, and at the end of the study, they're actually performing just as well as their wild-type counterparts. Now, it's hard to get too excited about this because what we know about the mice is they don't experience the same amount of neurodegeneration or cell loss as a human patient would. So even at the intervention, the 7 months, they still have quite a lot of cells in their striatum, so there are cells there to treat. We don't want to suggest that we can treat a late-stage Huntington's patient that's lost a significant amount of brain mass and cause reversal of the disease. But the good news is this suggests the cells are actually still there. Even if they're dysfunctional, we may be able to rescue them and restore them to health. And this is the results of the aggregates in the brain. This is an untreated or control-treated Huntington's mouse, and this is treated with the AAV RNAi. We almost entirely removed the aggregates within the striatum. So this was showing that we can rescue the neuropathology here as well. So what is it going to take for us to generate a clinical candidate for this therapeutic strategy? This therapeutic strategy really relies on two huge moving parts. One is the microRNA sequence itself. So we need that sequence to do two things. It has to reduce Huntingtin by about 40%. Because this is a non-allele specific approach, we're not going after 100% Huntingtin reduction. We think that would be a safety issue. So we're going about 40 to 50% of human, and rhesus monkeys are GLP tox species, so we have to prove safety first before an IND filing in a non-human primate. So our sequence has to recognize monkey Huntingtin, but also human, obviously. And it has to have low off-target potential, so really good specificity. We don't want to be knocking out any other genes. Obviously, that wouldn't be good, but also this therapeutic strategy is forever. You put the AAV vector in the brain, and it's going to be there for the rest of your life. So we want to make sure it's only knocking down Huntingtin and not messing around with any other genes in your genome, because obviously that could have downstream negative consequences. The other huge moving part here, in addition to the sequence, is the virus itself, the AAV vector. So we need an AAV vector to give us maximal striatal transduction in a large brain, predominantly neuronal tropism. That means that it predominantly transduces neurons. There are several cell types in the human brain. There's neurons and glia. Neurons are non-negotiable. If our virus can't get into neurons, we can't use it. And then a second point is that we want to have the potential for cortical distribution. So, in Huntington's disease, everyone used to talk about it as a disease of the striatum, but we now know that there's a lot of other brain areas involved in HD, and one other area is the cortex, and so we really want to aim to get both striatum and cortical coverage. So that's what I'm going to talk to you about in the next series of slides. So I really want to focus the rest of this talk on delivery. How do we get these things to the brain and to the areas of the brain that we want? So I think it's no longer a debate. It used to be a debate about where to treat the brain for Huntington's disease, but I don't think it's a debate anymore, that we really need to target the striatum and the cortex to have the maximum therapeutic benefit in patients. So there's a lot of studies that suggest that this is important. Imaging studies of HD patients show that there's early progressive loss of the cortex. If you reduce Huntingtin from both the striatum and the cortex in these animal models, you get the most therapeutic benefit. If you just reduce Huntingtin in either area alone, you get less benefit. So we believe at Genzyme that cortical and striatal targeting is going to provide the most bang for our buck, so the best therapeutic benefit to the patient. And we're going to try to rely on this axonal and transsynaptic transport of these vectors to really achieve that for us. Right now, it's not feasible to treat an entire human cortex with a virus because you do discrete injections. So unless someone's willing to sign up to have their brain poked hundreds of times over and have this virus delivered in hundreds of different locations, what we want to do is we want to inject the vector into the striatum, which is non-negotiable area, and hope that through transsynaptic transport, it gets transported out to cortical regions as well, because that would really be the home run. We have other challenges as well to translating this therapeutic to the clinic. Everything I've been talking to you about has been in mice. And if you can see, the mouse brain is about 1,000 times smaller and less complex than the human brain. So we really need to scale this up into a larger brain, which, you know, at this point in time would be the non-human primate brain. So what do we need our AAV vector to do in order to qualify as a clinical candidate? We set forth a number of different criteria. So like I said, we have to get maximal striatal transduction in a large brain. So now we have to go outside of the mouse model and move to something bigger. Again, we need to transduce neurons, and we are really hoping for that cortical distribution. So in the study that I'm going to talk to you about next, that was done in non-human primates, we looked at 2 different AAV vectors. Just like the RNA molecules, the AAV molecules also come in a lot of different flavors. And different flavors of AAV, or they're also known as serotypes, can do different things in the brain. So we chose AAV1 and AAV2 specifically to investigate in the primate brain. The reason we chose AAV1 is all the studies I just talked to you about in the animals were done with the AAV1 serotype. We know that AAV1 from previous studies we've done at Genzyme and others have done that we get both neuronal and glial transduction, so they definitely get into the neurons as well as the glia. And safety has been established for AAV1, so the very first approved, it's approved in the EU, AAV gene therapy drug, known as Collaborators, is an AAV1 vector. So that's really good rationale for why we should move forward with AAV1. So this is our lead serotype. Our backup serotype is AAV2. We haven't done any of the preclinical work in AAV2, but we do know that AAV2 is really good at transducing neurons in non-human primates and in humans. And there's probably been over a dozen, maybe up to 20 human clinical trials using the AAV2 vector. A lot of them have been done for Parkinson's disease. So we know that safety is not an issue with AAV2. So it's a really good backup serotype. So this is the study that I conducted. We wanted to evaluate the expression of these vectors in the monkey brain following an intrastriatal injection of either AAV1 and 2. We used male and female rhesus monkeys. It was a 30-day study, so we injected the animals with the AAV vector expressing, again, this green fluorescent protein, so we could see if it's getting to the cells and the areas that we want. We then collected the blood and the tissue 30 days later. And this study included not only AAV1 and AAV2, but it did include a control, which is an empty vector. It's just an AAV without any cargo. This just controls for the surgery, the injection, and the presence of this green fluorescent protein in the brain to make sure that didn't cause any issues. So how do we deliver these things? Well, we use a real-time MRI-guided convection-enhanced delivery to the monkey striatum to get these viruses into the brain. This is really important because this system, this real-time MRI CED system, is exactly what would be used in the clinic for the patients. We actually used a human grid and a human software and hardware, but just put it to use in the primates. So this was actually done in like a human OR suite at the University of University of San Francisco. Chris Bankevitz, a renowned neurosurgeon, performed the surgeries. And the real-time MRI allows the surgeon to make online changes. So before the surgery is even over, we know whether or not we hit our target. So we mix a little bit of gadolinium, which is an MRI tracer, in with our virus. So the surgeon can see where the virus is actually going, how well it's spreading. In real time, he can adjust. So if he hits a blood vessel and the virus kind of starts going elsewhere, he can adjust the needle. So really, again, before the end of the surgery, we have a really good idea of how well we got the infusion in that monkey. So here were the results, and I know most of you are probably not neuroanatomists, so you're not going to immediately look at this and be like, oh my God, but I will say that we got the results and we were absolutely floored and blown away. These are 2 AAV1 animals and 2 AAV2 animals. The brown is everywhere we got the virus, and What's important to note is the striatum, which is this area I'm outlining here with the pointer, that was completely covered with all the vectors. And we actually anticipated that because that's where we're injecting it. So it better be there, right, because we're putting it right there. If it's not, we got real problems. What was really exciting is that the vector spread from the site of injection throughout the entire cortex. So again, this is what we were really hoping for, is that when we inject into the striatum, because the striatum and the cortex are very well connected, that we could get spread throughout the entire brain. So this graph shows here that we're getting about 60% transduction of cells in the striatum. The AV2 was a little bit less. It was closer to 50% transduction. Here is just a higher magnification showing the frontal cortex and different layers of the cortex that were transduced with our vector. This is an overlay of AAV or GFP with a stain for neurons, and you can see that all of the cells that express our GFP also express this neuronal marker. So for both AAV1 and AAV2, we definitely transduced neurons in the brain, which was really important to show. And this is a 3D reconstruction of one of our AAV1 injected monkeys, where we took his brain scan and we reconstructed his brain, and we overlaid everywhere in his brain where we saw our virus or the GFP. And essentially, you can see the entire, almost 90% of this animal's cortex is covered with the vector. The other areas, obviously the caudate and the putamen, as well as other areas of the basal ganglia, like the subthalamic nucleus and the substantia nigra, are also very well covered with this approach. So this obviously begs the question, a lot of my colleagues say, well, you injected in the striatum, how the heck is it getting all over the place? And we know neuroanatomists have mapped the cortical striatal connections, and we know that the human cortex and the striatum, or the primate cortex and the striatum, are very well connected. There's lots of crosstalk between the two different areas. So clearly these two vector serotypes, AAV1 and AAV2, have the ability to move around from where we injected it to sites very distal. So just to summarize, the transduction patterns of both of our viruses following this MRI-guided CED delivery to the caudate and putamen suggest that these vectors can provide suitable distribution profiles for the delivery of HD therapeutics. And AAV1 is our suitable serotype for the Huntington's protein due to its superior neuronal transduction properties and the ability to transduce neurons and astrocytes. So obviously we're moving forward with this AAV1 microRNA approach, but there are several issues we still will have to contend with along the way. One is delivery to a non-perfect brain. So these animals, these are wild-type animals, right? This isn't a diseased brain. What's going to happen when we start injecting these vectors into a Huntington's patient brain, where things may not be entirely intact, connections may not be entirely intact? And so the only way to really address this before we go into patients is to conduct these studies in large animal models of the disease. So primate models of Huntington's, pig models of Huntington's, sheep models of Huntington's, and that's what we are we're talking about now, to see what will happen in the disease state if these processes of transport are still going to happen in a disease model. And then finally, always an issue is safety. We're always concerned about the safety of these approaches. One that I didn't really touch on is the off-target effects, obviously silencing non-Huntingtin related genes, but that's all about properly designing your microRNAs, which, you know, we can do bioinformatics and a lot of genomics to make sure that our sequence is very specific to the Huntington's gene. And then obviously the safety of silencing both alleles, that's always on everybody's mind. So we think that by doing a partial lowering, only lowering 40 to 50%, we will still see transformative benefit in the patient. I don't know if you heard about Ed's little, he said it's like wearing a backpack with bricks on it, right? The Huntington's patient has a backpack full of bricks. If you remove the backpack entirely, obviously that's ideal, but even if you just take out a couple bricks, they're still going to have a much easier time. So we're trying to take out maybe half the bricks in the backpack, and we think that can really provide the patient transformative benefit without concern over reducing the wild type, because we think having 50% of wild type Huntingtin will be fine, and there's definitely evidence to suggest that. So obviously I need to acknowledge my colleagues. This work that I talked about in the primates was done in collaboration with the neurosurgical lab of Chris Benkiewicz at UCSF, and it's a very big collaborative effort with my Genzyme colleagues. So be happy to take questions. Go ahead. I don't know who's. Is my lab in California? No, we're located in Boston, but our collaborators that do the surgical procedures are in San Francisco.
When you induce the transduction
Uh-huh.
of these vectors,
Yep.
viruses, do the viruses actually spread, regenerate themselves and build new viruses?
No, so they're not.
Just the ones that are delivered just get spread around?
Yep, the ones that are delivered get spread around. They're non-replicative. So that's a safety feature of the viruses. One reason why viruses make us sick is they multiply. So we've kind of taken that feature out. Hey, Chris?
You may have said this, when you saw the virus in the cortex.
Yep.
Was it in the deep layers that project the striatum? Was it throughout the cortex? And was it in neurons versus glia?
Yep, so that's a good, I didn't want to kind of get into the nitty gritty. I did see it very very robustly in cortical layer 5, which is the layer that directly projects. We saw it in both neurons and astrocytes, and it's interesting, the other areas of the cortex where we did see transduction, it was mostly in astrocytes. So lots of neurons in cortical layer 5, and then lots of astrocytes elsewhere. There were, definitely patchy. It wasn't 100% coverage.
What would you expect to be the timeline between now and starting human trials?
Yeah, so his question was about timelines between now and clinical trials. We're actually looking to file our IND next year, and I think clinical trials would probably be the phase 1. Again, we always have to start with the phase 1 safety trial. Would probably be the following year after that. Sure. So I'll answer that question. That's an excellent question. She asked about if there are effects outside of the brain, and I didn't show that data, but we looked at that in this monkey study. So I took organs from the animals. This was directly injected into the CNS, and we presumed that it would not escape the CNS because of the blood-brain barrier, but we have to know whether or not we're transducing liver, kidney, spleen. So we took all the organs from these animals, and we looked for our GFP protein and our virus, and there was none. So the vector stays within the central nervous system, which is a really nice safety feature of these vectors. And you had a second question? That's a really, really good question, and that is, I guess, the big downside of gene therapy. We cannot turn it off at this point. So you can turn it on, and you can get it in the brain and get it expressing. If a couple years down the road there are some untoward effects, unfortunately at this point in time with the technology we have, we can't shut it off. So that's why we are conducting very extensive and much longer than usual safety studies. So, I don't know if people in pharmaceutical industry know that most safety studies, 2 to 3 months, sometimes they're like 10 weeks. Our safety study will probably be a year long, which should give us a really good idea in monkeys if there's any deleterious consequences. So. But that is a good question, and it's always a concern for gene therapy. Yeah, that's a great question, too. So we actually, at Genzyme, we have an AAV Parkinson's trial. And in that trial, we're actually expressing, instead of knocking something down, we're expressing an enzyme called AADC. And we are able to see in monkeys out to 10 years, and that's the longest we've looked, the enzyme is still expressing. In human patients, we've seen it out to 6 years. And again, that's the longest we've looked. So theoretically, it should be there forever, for the lifetime of the patient. With the data that we have, you know, at least a decade, we've seen it.
Has there been any immune response?
That's another really good question. He's asking about immune responses, and that we looked at that again in the study. I didn't show the data, but we have not seen any immune response with AAV1 or AAV2. We looked for microglial activation. We did not see any, which is really good. Other vector serotypes, most popularly like AAV9, has shown some immunogenic responses, but with these particular 2 flavors of AAV, we haven't witnessed any in the brain. Anybody else? All right, thank you so much.
Thank you, Lisa. Well, can everyone just give a final round of applause to Lisa and Roberto and Wendy, and for finishing ahead of schedule. So I hope everyone enjoyed the presentation. I'm sure Wendy and Roberto and Lisa will be around. If you have any other questions, grab them during the rest of today or tomorrow. Enjoy your night, everyone.